welcome to Akebia Therapeutics Protect Topline Data Readout call. As a reminder, this call is being recorded. I would now like to introduce your host for today's conference, Kristen Shepard.
Thank you, and good morning. My name is Kristen Shepard, Senior Vice President of Investor Relations with Akebia. Thank you for joining us to discuss Akebia's top-line data from PROTECT, the second of our two global Phase 3 cardiovascular outcomes programs evaluating vadadustat for the treatment of anemia due to chronic kidney disease. We issued a press release containing this top-line data this morning. It is also available on our Investor Relations website along with the slides for today's call. For your convenience, an audio replay of today's call and the slides will also be available on our website shortly after we conclude today's webcast. Joining our call today are John Butler, our President and Chief Executive Officer, and Dr. Stephen Burke, our Chief Medical Officer. Before we begin, I'd like to remind everyone that this conference call includes forward-looking statements. Each forward-looking statement contained in this call is subject to risks and uncertainties that could cause actual results to differ materially from those described in these statements. Additional information regarding these factors is described in the forward-looking statements section of the slides for today's call and the press release we issued this morning, as well as in the risk factors in management's discussion and analysis sections of our most recent quarterly and annual reports filed with the SEC. The forward-looking statements on this call speak only as of the original date of this call, and we do not undertake any obligation to update or revise any of these statements. With that, I'd like to turn the call over to our CEO, John Butler. John?
Thank you, Kristen, and welcome. We appreciate everyone joining us on such short notice. In terms of an agenda for today's call, I'll provide some opening remarks, then Steve will provide more detail around the top-line data. As always, following our discussion, we'll open the call to questions. This morning, we announced top-line data from PROTECT, the second of our two global Phase 3 programs which evaluated vadadustat, our investigational HIF-PHI, as a treatment for anemia due to chronic kidney disease, or CKD, for patients not on dialysis. Before I get to the top-line results, I want to talk about our clinical program and vision for vadadustat. We launched our global Phase 3 program consisting of both Innovate and PROTECT because we recognize the significant unmet need of patients with anemia due to CKD and see a promising opportunity in vadadustat to advance the standard of care for these patients. Our global Phase 3 program consists of 4 large, well-executed studies of patients on dialysis and not on dialysis, some of the largest of their kind in the industry. To report data on 4 studies over a few months is a significant milestone. To do the work during a global pandemic is nothing short of remarkable. And make no mistake, we believe these studies have succeeded in providing us with extensive data sets in support of establishing vadadustat as an oral, once-daily standard of care for the treatment of anemia due to CKD. This is incredibly important, not only in terms of what it means to Akebia and our path forward, but also, and more importantly, because of what it means to patients. At Akebia, our mission is to better the lives of people impacted by kidney disease, and I'm very proud that we are moving forward toward that goal with data in hand that we believe supports vadadustat's approval. Our first global Phase 3 program, Innovate, delivered positive top-line efficacy and safety data for vadadustat in dialysis. With these clear results, we believe we have a straightforward path to submission and approval for a dialysis indication. And with approval, the significant opportunity to help the 500,000-plus adult patients on dialysis. We believe this translates to a potential multi-billion-dollar market opportunity in the U.S. alone. The top-line results from our second global Phase 3 program, PROTECT, have also been highly anticipated and are equally important as another step towards fulfilling our vision for vadadustat in non-dialysis. I'm pleased to report that vadadustat achieved both PROTECT's primary and secondary efficacy endpoints. The efficacy data was very clear and consistent with previous studies. Unfortunately, PROTECT did not meet the primary safety MACE endpoint. The MACE result is disappointing to all of us, and presents challenges to achieving our goal of bringing vadadustat to patients in non-dialysis. That said, while achieving the safety endpoint would have made our path here much easier and more straightforward, as it is in dialysis, we still believe we have a path forward in non-dialysis. We and our collaboration partner, Otsuka, believe this path is built on the totality of the data from our global phase 3 studies, including important key regional and patient subpopulation analyses from PROTECT that has been submitted for presentation at the upcoming ASN meeting in October as a late breaker. This data-rich presentation will allow us to share a more detailed view of our path to submit an NDA and secure approval for vadadustat for both indications at a scientific venue, which has always been our goal. Although the top-line data makes our path forward in non-dialysis more challenging, we believe our potential with vadadustat to address the significant unmet clinical need of patients with anemia due to CKD not on dialysis remains a multi-billion-dollar market opportunity. I want to stress that while we believe we have the opportunity to secure indications for both dialysis and non-dialysis markets, it's important not to lose sight of the fact that we believe each of these markets on its own presents a compelling multi-billion-dollar market opportunity for vadadustat. With data now in hand, let me remind you of our next steps. We are highly focused on our U.S. regulatory filing. Our teams have already started working on the NDA for vadadustat. At this time, we're planning for a pre-NDA meeting with the FDA before the end of the year, followed by our NDA filing for both dialysis and non-dialysis indications as early as possible next year. In parallel with these activities, we're continuing to work with Otsuka on the European filing for vadadustat. Over the near term, we're working hard to secure and prepare the full Innovate and Protect data presentations for ASN. We've already submitted Innovate for presentation, and as I mentioned, we just recently submitted Protect as a late breaker. If accepted, we'll be presenting these data sets in about 7 weeks at the ASN meeting. As well, we're actively working to secure publication within peer-reviewed journals. Beyond the work to move to filing, we are already supporting the commercialization of vadadustat in its first major market, Japan. All of these remain critical initiatives for us, and we're fortunate to be executing from a place of financial strength with a cash runway that extends beyond the expected U.S. launch of vadadustat. In closing, we believe we have a straightforward path to add a second commercial product in vadadustat to our renal portfolio for patients on dialysis upon approval. We believe we have a path forward in non-dialysis as well. The Akebia and Otsuka teams are continuing to prepare for the launch of vadadustat, if approved, for both dialysis and non-dialysis patients. I want to pause here and take a moment to thank all of our patients who participated in Protect and recognize our clinical investigators and their staff who worked harder than ever to close out these studies. Kidney patients are among the most at risk during this pandemic, and we're reminded of both the critical nature of our work and the significant need to advance care for these patients. I couldn't be prouder of the Akebia team for their work. I'll now hand the call over to our Chief Medical Officer, Dr. Stephen Burke, to discuss the Protect top-line results in more detail. Steve?
Thank you, John, and good morning, everyone. On behalf of the entire R&D team, I would also like to thank all the patients, investigators, and staff for their dedication to our mission. The Protect program was well-powered for efficacy and cardiovascular safety and included 2 phase 3 studies, Correction and Conversion, which collectively enrolled 3,476 non-dialysis dependent patients with anemia due to CKD. This was a very large, rigorous, and thoughtfully designed program to compare vadadustat to a current standard of care, darbepoetin alpha, an injectable ESA. Both Protect studies were global, multi-center, open-label but sponsor-blind, non-inferiority studies. In both Protect studies, the primary efficacy endpoint was non-inferiority of vadadustat versus darbepoetin as measured by the difference in mean change in hemoglobin between baseline and the primary evaluation period, which was between 24 and 36 weeks. The key secondary endpoint was non-inferiority during a secondary evaluation period between 40 and 52 weeks. The PROTECT program's primary safety endpoint was non-inferiority of vadadustat versus darbepoetin for time to first MACE in the combined PROTECT studies. MACE was defined as all-cause mortality, non-fatal myocardial infarction, or non-fatal stroke. The MACE events were independently and blindly adjudicated by the Brigham and Women's Hospital Clinical Endpoint Center. As shown in slide 5, in both studies, patients were randomized 1-to-1 to receive either vadadustat or darbepoetin. Vadadustat was initiated at a dose of 300 milligrams once daily, and starting at week 4 was adjusted up or down in increments of 150 milligrams within the range of 150 to 600 milligrams daily. Darbepoetin was administered intravenously or subcutaneously. Patients already receiving darbepoetin maintained their prior dose, and those on other ESAs were switched to darbepoetin and dosed according to the approved product label. Study drugs were titrated to achieve target hemoglobin of 10 to 11 in the U.S. and 10 to 12 outside the U.S. Slide 6 summarizes the patient's baseline characteristics, which were similar between the treatment groups and representative of the general CKD population with anemia not on dialysis. As expected, there was a high percentage of patients with cardiovascular disease and diabetes. Slide 7 summarizes the primary and key secondary efficacy endpoint data. In the correction study of patients without recent ESA use, PROTECT met the primary efficacy endpoint, demonstrating that vadadustat was non-inferior to darbepoetin. The difference in mean hemoglobin change was plus 0.05 grams per deciliter, with a 95% confidence interval of minus 0.04 and plus 0.15, with the lower bound not crossing the pre-specified non-inferiority margin of minus 0.75. The mean hemoglobin at weeks 24 to 36 was 10.39 for vadadustat and 10.35 for darbepoetin. The hemoglobin responses were maintained in the secondary evaluation period of 40 to 52 weeks. The mean hemoglobin at weeks 40 to 52 was 10.48 for vadadustat and 10.45 for darbepoetin, with a difference of plus 0.04. Again, the lower bound of the confidence interval was above the pre-specified non-inferiority margin of minus 0.75. In the conversion study of patients on active ESA, PROTECT again met the primary efficacy endpoint, demonstrating that vadadustat was non-inferior to darbepoetin. The difference in mean hemoglobin change was minus 0.01 grams per deciliter, with a 95% confidence interval of minus 0.09 to plus 0.07. The lower bound of the confidence interval was above the pre-specified non-inferiority margin of minus 0.75. The mean hemoglobin at weeks 24 to 36 was 10.77 for both vadadustat and darbepoetin. The hemoglobin responses were maintained in the secondary evaluation period of weeks 40 to 52. The mean hemoglobin at weeks 40 to 52 were 10.80 for vadadustat and 10.79 for darbepoetin, with a difference in hemoglobin of 0. Again, the lower bound of the confidence interval was above the pre-specified non-inferiority margin of minus 0.75. Having reported clear top-line efficacy results for vadadustat with INNOVATE, our phase 3 program for dialysis a few short months ago, the team is very pleased with these consistent and straightforward efficacy results for non-dialysis. Unfortunately, PROTECT's top-line MACE result did not deliver the same level of strength as INNOVATE's. As you can see on slide 8, PROTECT did not meet the primary safety MACE endpoint. The upper bound of the 95% confidence interval was 1.36, which was above the pre-specified non-inferiority margin of 1.25. The hazard ratio was 1.17, and the 95% confidence interval was 1.01 to 1.36. As John stressed, we believe the totality of the data from across our global phase 3 program supports the cardiovascular safety of vadadustat, particularly when considering the data within key regions and across certain patient subpopulations. We look forward to sharing a fuller review of the data at an upcoming medical conference. Slide 9 summarizes the treatment emergent adverse event data and the most common treatment emergent adverse events, those occurring in greater than or equal to 10% of patients in either group. These common events included end-stage renal disease, hypertension, hyperkalemia, urinary tract infection, diarrhea, peripheral edema, and nausea. The events were similar between treatment groups in both PROTECT studies. Consistent with the INNOVATE data, there were no Hy's Law cases and no difference between treatment groups in any liver-related safety parameters. In closing, I want to reiterate that we believe we have a very extensive data package in support of vadadustat's approval for the treatment of anemia due to CKD in both indications, and the team is already hard at work on our NDA. I'll now open the call to questions. Operator, we're ready for the first question.
Thank you. As a reminder, to ask a question, you will need to press star 1 on your telephone. To withdraw your question, press the pound key. Please stand by while we compile the Q&A roster. Our first question comes from Chris Raymond with Piper Sandler. You may proceed with your question.
Hey, thanks for doing this call, guys. I guess I get a sense from your commentary that there's an awful lot you want to save for the ASN meeting, but maybe I'm just hoping that you can put a little bit of meat on the bones, I guess, in terms of this commentary on subpopulations in key geographic regions. Is the premise here that there will be some subpopulation perhaps ESA naive versus having been exposed to ESAs, or is there some other subpopulation you're thinking that will become completely obvious at ASN, or is it maybe just any sort of color you can get as to what that will look like when we see the data at ASN? Thanks.
Hey, Chris, thanks so much for the question. So we really recognize how important it is to present this data in a scientific session. And again, let's step back. What we talk about here is the totality of the program, right? We have a global program with 4 different studies across both dialysis and non-dialysis, first and foremost. We have an efficacy story that is outstanding, extremely clear. The dialysis indication, clearly a straightforward path. PROTECT, not as straightforward. No matter what we would say to you today, you can't get away from missing the primary safety endpoint. But when you look at that totality of the program, including these other analyses, which is absolutely critical for us to present at ASN, you see a path forward. It's a clear path forward. But Chris, beyond that, we have a path forward, right? We've identified that. But this is an incredibly rich data set, right? So over the next 7 weeks, we get to do further analysis to continue to strengthen that data set and deliver that all. ASN isn't 6 months away, right? It's less than 2 months away. And giving you the complete story at that point makes a lot more sense for us. And it's the best thing for the product going forward.
All right. So I get that. So I guess just to be clear, so the path forward is only incorporating the existing data set. Is that correct?
That's absolutely correct. Yeah, it's our expectation is we'll be filing for both indications with the current data set.
Okay. All right. Thank you very much.
Thank you. Our next question comes from Duffy Yang with Mizuho Group. You may proceed with your question.
Hi, good morning, and thanks for taking my question. Just a couple here. With regards to the MAA submission, would you walk us through the strategy there? Would that be contingent upon the FDA regulatory pathway? And secondarily, with regards to today's data, how do you view the potential implication to vadadustat launch in Japan?
So the first question, Difei, was around Europe and the MAA, and that strategy hasn't changed. We are supporting Otsuka on that MAA filing, and again, it's nothing has changed in our approach to Europe. The second question was around the launch in Japan. And again, there's no change there. They are actively launching the product, our partner MTPC, in both indications. We're doing everything we can to support them, and I know their expectations for vadadustat are high.
Thank you, John, for taking my question. Just a quick follow-up. Do you see a scenario with regards to the subgroup analysis in which you think there may be a possibility to do additional trial to solidify that data analysis?
Right. So so we look at the totality of the data, including these key regional and patient subpopulation analyses from PROTECT. We think when you see that data together, you'll agree we have a path forward. Not a straightforward path forward, not an easy path forward, but when you look at the totality of the data, we think it supports approval in both dialysis and non-dialysis.
Okay, thank you, John.
Thank you. Our next question comes from Eric Joseph with J.P. Morgan. You may proceed with your question.
Hi, thanks for taking the questions. John, just interpreting your comments about, regulators looking at the totality of the data, does that suggest an integrated analysis of MACE across both the non-dialysis and dialysis populations? Have you discussed that type of integrated analysis with both regulatory bodies, FDA and EMA? And also just on the regulatory path forward in Europe, what's your sense of whether that path in the non-dialysis population is more or less challenging than in the U.S. with FDA? Thanks.
Thanks, Eric. So the analysis of the data was always going to be about the totality of the data, right? Every FDA meeting we've had as we were designing this was talking about the totality of the data. I know we all focus on kind of one number here. That's not the way the FDA does drug reviews. So it's always been about looking across the entire program. They've always preferred to see dialysis and non-dialysis data together, and so given, very pleased with how we designed these studies. We did this, and I think we've talked about this many times, Eric, about how we designed both the dialysis and non-dialysis programs extraordinarily similarly, so these kinds of analysis could be done. This was always our expectation, and that's the way we're thinking about that. Now, we are, as I mentioned, we're planning to have a pre-NDA meeting before the end of the year, and that's where we'll have the opportunity to lay out our strategy to the FDA. Obviously, they won't be opining on data, but we'll talk about the strategy forward. We don't really see a difference from a strategy standpoint with the EMA. And again, Otsuka is leading that filing, but obviously we're working very closely together on it. But we see the same path forward for both dialysis and non-dialysis.
Okay, great. Thanks for taking the questions.
Thank you.
Thank you. Our next question comes from David Lebowitz with Morgan Stanley. You may proceed with your question.
Dave, I'm sorry, we can't hear you.
Can you hear me now?
Yes.
When you compare the hazard ratio on the non-dialysis to the dialysis population, it seems to be quite different with the dialysis
David, you're breaking up a lot. I know you were asking about the hazard ratio difference between innovate and protect, but beyond that, I didn't get the question.
I just was asking why the hazard ratio on innovate and protect is so much.
Why is there a difference? I think the question is.
Okay, yes. We'll try to answer the why question, which I think is what you're asking.
Yes.
I don't know that the data is what the data is. So we are actively investigating that. As John alluded to, some regional and other patient characteristic factors that we'll discuss at the ASN. I think as John mentioned previously, it will be clear when we make the presentation at the ASN, why there are some differences.
And ultimately, they are different patient populations, and that's why you have to look at them as separate patient populations. Again, we think the totality of the data is incredibly important, but we also believe that you have to look at this as two different indications, and that's how we're going to be submitting the NDA.
Thank you for taking my question.
Thank you. Our next question comes from Ed Arce with H.C. Wainwright. You may proceed with your question.
Hi, everyone. Thanks for taking my questions. I recognize, as you said, John, the importance of presenting the full data set at ASN, but I was just wondering if there is any way you could today give us a little more perspective on the data itself. In particular, it seems to me anyway that the AE rates in the conversion study may have been what is driving a slightly higher or quicker MACE rate. And perhaps if you can discuss perhaps any differential among the three components of MACE, either in correction or conversion. Thanks.
Thanks for the question, Ed. So I don't think you can read anything from the AE tables to MACE. There's really, if you look at those AE tables, there's really no difference between the treatments, with the exception of diarrhea, which we've always seen a little more with vadadustat. But beyond that, I kind of completely appreciate the desire to understand more of this data and more of what we're seeing. But we think it's critically important to do this presentation at the ASN meeting. This was always about top-line data, and that's what we're sticking with at this point. We are fully confident. Again, I'm not trying to convince anyone of anything. What we're trying to do is communicate to you, here's the top-line data. We recognize that missing the primary safety endpoint makes our path forward more complicated. Frequently, that would be there's no path forward. As we've looked at the data, we see a clear path forward. Not a straight one, one that needs a lot of work. It's based on the totality of the data, the strong efficacy result, the clear result in dialysis, and the subpopulations and key regional analyses in PROTECT as well. We think when you put all of that together, that gives us a clear path, and that's why we're confident we're going to file the NDA in both dialysis and non-dialysis, and we hope for approval. 7 weeks from now, we'll have a much fuller presentation. We're actively working on getting these studies published as well. We submitted the late breaker to ASN yesterday. We'll see whether or not it's accepted. We hope it will be, but you'll get a very full read of the data in just a number of weeks.
Okay, great. And then perhaps just one quick follow-up. Given that pre-NDA meeting by the end of the year and your discussion with the agency then about your strategy, do you think you'd be in the position to disclose any sort of agreements or challenges coming out of that meeting?
And we always disclose appropriately the way we should. If something comes from that meeting that is material around our path forward, or different from what we've been telling you, we'd certainly let everyone know that. If it's, again, I mean, it's about their review of the data in the long run. So, that's would be my expectation, that they'll understand what we're trying to do and look forward to filing the NDA. But if something changes in that meeting, we'll certainly let everyone know.
Great. Thanks so much.
Thank you. Our next question comes from Bert Hazlett with BTIG. You may proceed with your question.
Thanks for holding the call, and thanks for taking my question. Let me try to come at it a little bit differently with regard to preserving the discussion at ASN. Could you just remind us of the geographic differences between Innovate and Protect? I've got one or two more.
Yeah, this is Steve. Yes, we had U.S., Europe, and rest of world, and those are stratified, and half of the patients were U.S. and half were ex-U.S.
In Protect.
In Protect. In Innovate, it was a little more skewed towards the U.S. 60-40.
60, yeah.
Okay. So, thinking about this, projecting forward, assuming there is a path forward with regard to non-dialysis, how do you think this result ultimately affects your positioning in the marketplace, assuming you're able to work through a regulatory licensure?
So, as we've talked about a lot in the past, Bert, this is a huge market opportunity, right? This is a multi-billion-dollar opportunity. In the U.S. alone, I think $2 billion in dialysis, $3+ billion in the non-dialysis market. As we've always said, there's room for multiple competitors, and that hasn't changed at all. Our perspective hasn't changed at all. There's a high unmet need in the non-dialysis patient population. Most patients aren't treated or not treated appropriately. There's great room for growth there. But dialysis, we've talked about the opportunities there as well. We think it's very significant. We have a great partnership with Vifor Pharma, and with TDAPA in place. We think that's really going to drive adoption. So we really do believe that we're well positioned in both markets. And of course, don't forget that in the non-dialysis population, just the fact that you're delivering vadadustat as a once-a-day oral product, that really alone could lead it to be a new standard of care, and it differentiates us from certainly the injectable ESAs, but other HIF products that are ahead of us as well.
Okay, thanks. I'll leave it there. Looking forward to the ASN data. Thank you.
Thanks, Bert.
Thank you. Our next question comes from Kenneth McKay with RBC Capital Markets. You may proceed with your questions.
Hi, thanks for taking the question, and condolences on the MACE outcome there. Maybe from the preliminary analysis, did anything stand out in the composite MACE endpoint as driving inferiority versus Aranesp among, again, the composite of multiple endpoints there? And did you see any difference between correction and conversion? And then my second question is, did the Aranesp MACE events, the Aranesp arm MACE events, accrue as expected versus historical trials? And I'm asking that just because I'm curious how this outcome might sort of impact the class, given competitor trials hadn't used Aranesp in this population as a control and had used a placebo. Thank you.
Okay, that was a lot of questions there, Ken. So first, I appreciate the sentiment, but we don't think condolences are necessary. It certainly would have been much more straightforward for us if the primary safety endpoint had hit. But again, we do believe we have a path forward here. There was nothing remarkable from an overall MACE perspective outside of kind of what's normally seen, Steve, right?
Yeah, the trial ran as expected, really. The way the events accrued, there's no dramatic thing to tell you.
Yeah. And again, components, differences between studies, et cetera, all of that will be part of the ASN presentation or the publication. Obviously, the presentation can only cover so much ground.
Gotcha. Thank you.
Thank you, and I'm not showing any further questions at this time. I would now like to turn the call back over to John Butler for any further remarks.
Thank you, Josh, and thanks everyone for joining us today. Hopefully you heard the clarity we have around the totality of our data, the strong data set we have between Innovate and Protect, the excellent results we've gotten across all the studies in efficacy, the straightforward path we have in the dialysis indication, a multi-billion-dollar indication on its own. And while we're disappointed in the safety result for Protect, we see a clear path forward. And we're very excited about the opportunity to present this data to you and to the physician community at the ASN. I think that'll probably be the next time that we speak to you, and we're looking forward to that. Thanks for joining us today.
Thank you. Ladies and gentlemen, this concludes today's conference call. Thank you for participating. You may now disconnect.